Also, thank you everyone for attending my webinar. So also it's my great pleasure to have this opportunity to present some of my our work on the humanized animal model. So my topic is improved translation by using humanized animal models for immunotherapy. In today's presentation, I'm going to talk about some translational challenges in immunotherapy. Also, I'm going to present human immune system reconstitution in biocytogen generated immune deficient mice. We call it BNDG mice. And also, I'm going to talk some application of biocytogen humanized checkpoint cytokine mice for immunotherapy. So, loss in translation can occur in every steps from basic science to clinical research, especially between the basic science research to clinical phase 2 or phase 1, phase 3 trials. So about more than 80 to 90% of drug development stopped during this time period or this stage. So we call this stage is the valley of death. A bunch of reasons related to this mode of in translation. So a critical effect is about the animal model. So there are a lot of immunological difference between mouse and human, as indicated in this publication. As I summarize, so the different percentage of immune cell populations between the mouse and human. Like, for example, the lymphocytes in the mouse is about 75 to 80%, but in the human is about 30 to 50%. And for neutrophils is about 10 to 25 in the mouse and about 50 to 70 in the human. So this difference may cause some different response to some stimulus. And also there are some different immune response, like a Th1, Th2 response in mouse and human. In mouse could be Th1 response. In human could be Th2 response. Also there are different biomarkers on the immune cell surface. Some biomarkers are absent in the mouse, but also present in the human. Also, they have a different FC receptors, including FC gamma receptors, or different subtype of immunoglobulin, or different function like response to some in some disease condition. For example, in mouse, some drug could show the inhibition, but in human could show the activation, and the more other difference. All these difference can cause some translational problem. And currently we use a lot of some animal models for cancer study, like a PDX model, CDX model in some immune compromised animals. Also some genetically engineered mouse model, also that we call the GM model, was GM derived GDA model in some immune competent animals. But compared to the clinical trial or in human conditions, all these models, there's really not relevant to human conditions. That includes some like the immune system is compromised or immune system. Tumor microenvironment is genetic or androgenetic. The human relevance is really variable. So the results from these animals is very difficult translated to the human condition.